So my job was to give you a very brief overview of the field. I've taken the stand that most of you know enough of the field to want to have something a bit specific, and my own standpoint is a little bit of the problem of reproducibility and how we make sure that the results are robust. And so we're going to talk a lot on the effect sizes specifically. But I will start with some motivation. I will look at a quick background of imaging genetic, of genetic variation, just in case some of you are not up to speed with that, and then give you a little bit of a, what happened in the field, and it's going to be a very biased and very sparse review of this, but hopefully you'll get something of it. And then, most importantly, I think I will try to move to a little bit of a practical, how do you compute effect sizes when you look at a PDF of a paper and you want to know what's going on really, what kind of actual effect size do I have in this study, and then a little bit of the future. So first of all, the motivation. I think everybody should be interested in the field. The geneticists, they want to understand the gene, and knowing the impact of the gene on the brain is one way of doing this. And obviously the neuroscientists want to know if, let's say, if the dopamine gene is impacting a specific brain network, then that's interesting to understand the brain. And the psychologists and the cognitive behaviorists and so on, they do need to understand behavior from all this information. But most importantly, to me at least, is that it has a great promises for personalized medicines and predictive medicine. That's if we were able to say to some person that let's say you need to probably actively work on that sort of thing to prevent some disease in the future from this information, that would be a good thing to have. And imaging genetic is there's no great model, animal model for the human brain. I mean, mice don't speak and so on. So the field itself is really exciting and important in that respect. And it's illustrated very well in the in Andreas Meyer-Lindberg review of 2006, where you see, for instance, the COMT variation. We know that it has an effect on the prefrontal cortex. We know that prefrontal cortex is involved in working memory. We know also that it has some link with schizophrenia. So linking the genetic information to the phenotypic or clinical phenotype and through the brain, for the brain system, for the neural system, may give us some mechanism aspect to how it works. So that was the promise, and that still is the promise of brain imaging genetics. It's also important that people would think that maybe because the brain is actually closer to the gene, even if our brain phenotypes are actually very far from the genetic activity, the genetic aspect of because they're still closer than the cognitive behavior phenotype, then maybe we will have a more penetrant aspect to the study. So that's those things were very exciting and are still very exciting, and I'm going to review some of that in the talk. A quick background on genetic variation. We assume at that course that you probably have some background already, but just to remind you that there are many variations in genetic variation, from the one SNP, from the very one change of one nucleotide to a very large deletion in a chromosome. All those variations will have some, may have some impact, and obviously the larger the variation, the more likely the impact will be. So I'm going to just quickly review two of those variations to make sure that everybody knows about that. One is the SNP, the single nucleotide polymorphism, where a single nucleotide is changed. So there is, let's say in the population, 70% of the population has a C on that specific locus in the genome, and maybe 30% of us have a T, which has and possibly that change is making some functional change in terms of in terms of the actual function of the construction of the protein and so on. So that's a very simple polymorphism, that's a very simple variation that is very, very well studied and very often studied in our field. The second very classical and somehow very common study of genetic variation is the CNV. So the copy number variant is either a deletion or a duplication on a gene, and that duplication can happen on one chromosome or two chromosomes or zero chromosome. Right? So that and that can be a deletion, that can be a duplication. These variations have often a fairly large impact or not, and we don't know, and that's all the study of the copy number variant aspect. So that's two very simple variation that I would like just to remind you for the rest of the talk. So a very, very quick and biased history of the field. I think, first of all, the field started with one variant, let's say a SNP, let's say some variation on a gene, and somehow small cohort size. And I'm going to switch now towards more like some technical aspects on how we make sure that we compute the effect size and those things. Then we had the possibility of looking at across the whole genome, or with the GWAS, with all the chips that we were able to capture thousands, hundreds of thousands of variation on the genome. And that led to GWAS with sometimes small-ish kind of cohort, and then larger and larger cohort, and we will see that. Then there was some interesting work, trying to see, hey, we have with a GWAS, we're looking at the effect of a SNP on a particular brain phenotype. Why don't we look at the effect of a SNP on any phenotype? And if we have 3,000 or 5,000 or 1 million SNP, why don't we look at the effect of million SNP on 50,000 brain voxels, for instance? And that led to the genome-wide, brain-wide studies that I will. And then the question arises on, can we use multivariate studies and look at what is the effect of several SNPs or several variations on the genome on several phenotype brain imaging phenotype. And that led to the multivariate aspect, and I will review very quickly a couple of those things. I will pass very briefly on the heritability. Heritability came as not only a field in itself, but also something that would be useful for any studies that we'll be doing in imaging genetic. And then I think more and more the field is moving toward the study of networks, and that is also of a great interest and great promises. The interpretation is absolutely key. I think Jason has a whole talk on the interpretation, and I think that's probably the most important talk of this morning. So let's look at a very first and very interesting and thought-provoking study published in 2002, I think, by Hari et al., looking at the polymorphism, the 5-HTT serotonin transporter polymorphism, where we knew that the short allele, the people who have either a short allele or long allele on that polymorphism, and we knew that of that variant, and we knew that the short allele people are a bit more stressed, and they also seem to have more activity in the amygdala when they're presented by fearful faces compared to non-fearful faces. So that was an extremely interesting study where you would link both the behavior and looking at the effect of that variation on that serotonin transporter with some explanation of the mechanism. Interestingly, the effect size, so the author report that they have one group with the short allele versus the other group with the long allele had a very the mean of the effects of the fMRI signal on the amygdala was about 0.28 for one, 0.03 for the other one, with some standard deviation of the mean. And that's what we get in the paper. We get those means, those standard deviations, and the number of subjects. And you can see that there is very few subjects in those very early cohorts. There's only 14 subjects that were scanned and interestingly as well, this study was actually replicated on another 14 subjects. So how do we compute the effect size? What's the actual effect size, and what is the effect size in that study? So the effect size in general is just, let's say you have two means and you want to look at the actual effect size. It's just the difference of the mean, right? It's how big is the difference between those two groups. What is the standardized effect size? And you probably have seen Cohen's d in the literature. What is the Cohen's d standardized effect size? Well, you take the standard deviation of the data, not the standard deviation of the mean, the standard deviation of the data, and you normalize by the standard deviation of the data. And what is the effect, the z effect, which is has a one-to-one mapping with the p-value, right? It's almost the same thing. What is the z effect? The effect, it's simply the effect size divided by the standard deviation of the mean, which involves to have and the more data you have, the smaller that standard deviation of the mean will be. So you divide by square root of your number of subjects in general. So that's just reminding you those basic things before we go into some examples in epigenetics. So let's compute that effect size. I've got the standard deviation of the mean. I know now that the standard deviation of the data should be about square root of the number of subjects, -1 if you account for this estimation of the mean and times the standard deviation of the mean. And then of course, if you have two groups with two sigmas, then you have to recombine those things, and that's not difficult, and you can find it easily on the web, or you can actually write it from scratch, and it's not difficult to find what's the good formula to combine those two standard deviation of those two groups. And then you look at the effect size, the Cohen's d effect size of that study, and you find that the Cohen's d effect size is simply a number of subjects, square root of the number of subjects, and the standard. I mean, you get the sigma, and then you get the effect size, and then you find the Cohen's d effect size. And the Cohen's d effect size is 1.05 in that study, according to my computation and others' computation. Let's keep that number in mind and go on. What's now the percentage of variance explained? How do you go from that and say, hey, how much of that variance of the data could that explain with this effect? Well, you have to be a little bit more skilled in writing models and things like that, but it's easy, and I'm very happy to show you exactly how it's going on after the talk. If you want to talk to me, and we can take a piece of paper and do it. But you can write that model, those data, and as a function of the difference of the mean of those two groups plus some noise, right? And if you look at the total sum of square, and then look at what's going to be the variance explained by the difference of the means, you find that the variance explained is something like about 40% of the. And that's a very large number. Even if you have already done some of those computations before, you know that's a very large number. So there's something that we should be, questioning ourselves. Hey, 40% of the variance explained, is that something reasonable? And I want to really go back, because in the history of any field almost, we had this sort of like questioning to start with. So let's move on and say that now we've got a chip which has maybe millions SNP on the population, and that was I think probably one of the first GYs done on 733 subjects by Shen et al. On the ADNI data. They were looking at the brain imaging phenotypes, like a volume of the brain structures, and look at the effects of all those SNPs on those brain structures. And because we have to correct for the number of comparisons, so now we've got 1 million comparisons. I think in that case there was not as much. I think it was more of the probably 500,000 SNPs. We have to correct for the fact that we're doing all those tests for each of those brain regions. And here in this instance, the usual threshold that people apply is 5 to the minus 8. And that's a usual G-wise threshold that you will find in the literature. It can be adapted depending on various things, but that's a kind of classical number for that. The only actual very strong effect was the APOE4 with this in this cohort in ADNI, which is the Alzheimer cohort. That sounds very reasonable and very reproducible, and that's a very strong result. The other SNPs that came close to significance or were interesting, it's unlikely. It's probably unlikely that they will be replicated because of the and one aspect of that study is that the number of subjects is actually quite small for a genetic study. It's very big at the time for an imaging study. In 2010, it was studies were more of the size of 40 subjects, 50 subjects, maybe 100 subjects. So it's a very big study in that sense. For an imaging study, it's a very, very small study, almost ridiculously small for a genetic study. And again, it will depend on the effect size. So let's say we want to compute the effect size of the APOE4 variant. So we've got 733 subjects. So again, it's a small study for genome-wide association study, and only the APOE4 is confirmed, I think. And the p-value of that SNP was 6 to the minus 10. So how do we go and compute the effect size? Well, I think now we have more ideas of how we do that. We go from the P to Z is kind of easy. It's called the inverse survival function. You will find that in any package, in any language. You can compute as well as you can compute the p-value for Z value. You can compute the Z value for a p-value. There's a one-to-one mapping for that. So you compute the Z that is corresponding to the P, and that's an easy thing to do. It's one function. And then from the Z, you go to the Cohen's D. And the Cohen's D, we know now that it's just, it has a factor of the square root of the number of subjects. And the Cohen's D will be 0.2, about 0.2. So that's, again, that's an interesting. I'm trying to give you a sense of the amount. What sort of value should we expect, and what sort of value should we have? And that's to me that's a clear, very important sort of reaction that you should have when you read a paper, and you should be thinking, okay, I've got a very strong p-value. What's the effect size? What's the confidence interval? So I leave you for a little exercise, and we can talk later on that. What is the power for that p-value and effect size for the number of subjects? That's, power is key in making sure that those results are actually solid. And I'm going to give you a couple of effect sizes again in the literature. First, on the BDNF, on the MED-VAL variation on the hippocampal volume, has been in a meta-analysis by Homologic in 2012 has been computed as being Cohen's D of 0.13. There's the JSON studies with a very large cohort looking at the mark of the hippocampal volume as well, and looking at how much of the hippocampal volume was changed by a risk allele, and that was 0.6%, so a bit more than 0.5%. That's the kind of effect size that we should expect. There's the effect size of intermediate phenotype are not very much greater than others, and that's a very important message to keep in mind. The people would think that because you were looking at brain imaging phenotypes, the effect size may be bigger than the actual clinical or behavioral or phenotype. That's not always the case according to Flint and this review. And he looks at the mean odd ratio for a population of cohort of disease versus a control population, and that odd ratio on average is 1.15. It's a small difference of the small odd ratio. And the variance explained by a locus is about 0.5%, as we've seen in a JSON study, or even less sometimes. And some biological effect sizes that are much closer to the biology of the cell are actually smaller than that. For instance, 0.1 or 0.3% for some protein concentration or something like that. So it's not clear that the further away from the cell you are, the smaller the effect size. That's not clear at all. And there's the more recent study by Franke et al. On again, the volume of hippocampus. We're looking at how much of the volume is actually reduced by a risk allele or by the SNP allele, and that's about 0.7%, according to what I could see, with an odd ratio of 0.94 for schizophrenia versus control. All those numbers are visible. And that's, if you look at, I don't know if that's very clear, that's the percent, oh, I've got this mouse now. That's the percentage of variance explained, and you see that none of those variants are greater than 1% for all the brain phenotype, but also for some other phenotypes or clinical phenotype. So some studies you will find, and I think you will still find in the literature studies that are very big effect sizes. So you should always wonder whether those effect sizes are reproducible. So this RA study with 40% of the variance seems too big. There's the COMT and DBLF, DLPF, the dorsolateral prefrontal cortex meta-analysis by Mir, which shows that the effect size is 0.5, but most of the studies have very, very small number of subjects. There's this gene and the cortical area, which looks at the Cohen's d of one, which sounds to be like a very big effect size as well. Let's move on, and think that the effect size is one thing that we should really look at and very, very be careful. How about multivariate approaches? There are several possibilities. I'm just going to give you two very brief overview of those. One is to look at some pattern of a multivariate pattern and some weights on the subject on those patterns. So ICF, for instance, would give you that kind of information. How much a subject is related for that pattern of activity in the brain or that pattern of SNPs in the genome. And if you do this, if you do the ICA on the genetic side and on the brain imaging side, you can look at the correlation of those patterns, of those weights per subject. Let's say that there's a pattern, a genetic pattern that has some weights on all the subjects, and those weights will be correlated with the brain imaging pattern on the same subjects. That's Vince Calhoun's approach, and he's developed a lot of those approaches, and it's an interesting way of looking at things. Another approach is to think, okay, let's have a brain phenotype and have all those SNPs as let's say thousands of SNPs as regressors in a big linear model. And because you have so many regressors, you need to say, hey, I can't actually fit all those things because my degrees of freedom are too small. I don't have as many subjects or more subjects than I have SNPs. So I've got that long series of regressors in my linear model. I can't actually, I can fit, but there will be zero error. So then what you do is you introduce some constraint on sparsity. You say hey, I want something that gives me the best fit, but also has a very sparse solution, so pick up only a few SNPs. And you can do this, and that's led to lasso and all those techniques in linear regression. And you can do that for a multi-variate aspects, where you will be looking at all those brain regions and looking at all those SNPs that have some sparse solution for the impact on the brain regions. There's very interesting studies in this line of thought by Vunu and Tom Nichols and others, where you look at what are my best pattern in terms of coefficient for the SNPs and in terms of the genes, the phenotypes that are explaining best all my data. And that's led to the sparse regression, rank regression, sparse reduced rank regression techniques and so on. So all those things will give you two things. They will give you a pattern on the brain, which are weights on your phenotypes, weights on your brain imaging, and a pattern on the SNPs or the genes, and you would say, hey, those two patterns seem to have to be related in my cohort. And that's the basic of those multivariate techniques. They give you two patterns, two series of weights, one on the brain imaging side, one on the genetic side, and you would say that those things are related. Then you have to assess what's the significance of that relation. Then all those effect size aspects are important to rethink. How do we assess how big is the effect, not only in terms of the p-value, but and you have to think of how you interpret those things, and do more statistics to elicit those brain regions and those SNPs that are actually involved in those patterns. So that requires a lot more statistics and a lot more computation and also a lot more interpretation to be done at the end. Quickly, heritability is the amount of genetic variants, of the variants of your phenotype that is explained by the genetic information. I won't describe that because I think that's going to be described further in the course. But having a good knowledge of what we should expect in terms of heritability of our brain phenotype is important. Usually they are fairly heritable. This is a study by Roberto looking at and most of the phenotypes have a 50%, 60% heritability, all those brain volumes in the substructural brain volumes. And then moving to, because I see that the time is a bit running, moving to networks. I picked up that study, which I thought was interesting. It's looking at the fractional isotropy phenotype, and these studies actually look at what's the effect of a SNP on the whole fractional anisotropy phenotype. So what's the difference between the population that has one or two allele on that SNP and versus zero allele on the brain imaging phenotype. And they look at they actually created some networks saying, oh, those two SNPs that have the same sort of the same sort of impact will be linked in a network. And they create networks of SNPs, and they can then look at whether those networks of SNPs are actually relevant in terms of networks that we know in terms of genetic networks. So that's very interesting. And then looking at those hubs of those SNPs that are actually very well connected to other SNPs. What are the pattern of impact that we get on the brain imaging from those? And you see that there are some SNPs that will have some impact on many brain regions, like a pleiotropic aspect of the SNP, and there will also be some brain regions that will be impacted by any of those selected SNPs. So that's it's just a new look. Again, I think we have to be very thinking very hard, but what is the exact interpretation of those? How do we make sure that we understand the effect sizes? What's the variation of the fractional isotropy per risk allele and so on? I think those things are still missing in these early studies. So, imaging genetics, to kind of start to wrap up. There's a combination of problem that may occur, and it requires a lot of effort to make sure that on the brain and on the genetic side we have a good data sets and a good preprocessing. The large number of subjects that we need for genetic analysis are often difficult to get from on the brain side, on the MRI side. That is changing slowly with all those big projects like the UK Biobank, the IGNIMA consortium, and that's how things are being done now in the field. That we've really moved from small studies of small ends to very large ends, which requires different kind of approach, a lot of more collaboration, a lot of more open data being available. There's a lot of multiple comparison issues that I've talked briefly about. So if you look at both the SNP and the voxel level, you will have to have such strict p-value that you're very unlikely to find a significant effect. There's also a lot of flexibility in analysis on both sides. Like you may be able to retry and this flexibility in terms of the models that you can apply on the imaging, but also in terms of how you pre-process and how you what are the aspect of the genetic aspect that you'll be looking at. This flexibility has a danger, which is called the p-hacking danger, and the p-hacking danger is, if you retry and retry, you will eventually find a significant effect because just of the effect of retrying on the same data. And it's very easy to do this, and it's very common to do this, unfortunately. So there are some trend, and I think it's a good trend for people to actually pre-register exactly what they're going to do and exactly what they're going to test and how they're going to test and how they're going to do it. And that is protecting us from the p-hacking problem. That is a very common and very difficult problem to solve, actually. There's also the aspect of the trendiness of the field and the capacity of the human brain to rationalize any result that we get. I was told that any SNP, any gene that you find, you will find a possible explanation somewhere of why it has an impact on the phenotype, and it's very easy. And again, if you give a cognitive scientist a noise image that looks like an fMRI pattern, you will find that this pattern is interpretable, and maybe the subject is doing this and this and this. Right? It's so easy to rationalize results because we just want those results to be true, right? So that's something that I think we have to be aware as a community, and this pre-registration and looking at very maybe resampling techniques to look at the variation of the p-values and so on would be are good things to do. So conclusion, you probably have read, or if you haven't, you really should read the UNEDIS 2005 paper, which is untitled, Why Most Results Are False, Why Most Research Results Are False. And UNEDIS, and it has a good model to explain why that's the case, and power is definitely one problem, the number of subjects is definitely one problem. And you should read those, why, in which field, and why those, there's a possibility that the results are not solid. I think all those rationale actually apply in the imaging genetic field. So we should. This is my conclusion. Look at the effect size. I've concentrated a lot on the effect size. I hope you get now a little bit of way of knowing how to compute those effect sizes. And if you don't, I will put some more material on the web to make sure that that's the case. But look at these two studies, for instance, looking at the effect size either on BDNF and hippocampal volume or COMT on the dorsolateral prophoton. So those are meta-analysis of looking at all those studies that have studied this specific link between the genetic variant and the brain phenotype. And on the x-axis, you've got the year of publication, and on the y-axis, you've got the effect size. And on the top, for the top one, the size of the circle is actually the size of the cohort. And you see the trend that there is a small cohort with big effect size, which is called the winner's curse effect, and then larger cohort and less effect size. And those things are really important to remind ourselves when we're doing a study that is definitely a need for replication and therefore open data to get that happening. And I think that's my last slide. What's next? We need to make sure that our effect sizes are reasonable, that we have a strong reproducibility, and then again, all comes back to the interpretation in a way that we don't p-hack, we don't make something interpretable when it's not. So going into reproducible science is a hard path, but it's a necessary path, especially in imaging genetic. Thank you very much.
Great. Thanks, JB.